Happy Friday, biologists. Today is April 10th, 2020. We are going to be continuing talking about hybrid crosses. So we're going to be talking about crosses, so how we get different traits from different parents. So we're still on the same standard of traits being passed down. Today our objective is to perform and analyze dihybrid crosses by completing problems. And as always, there's my contact information. If you have any questions, please continue to contact me. So we are on the final day of your packet. So now we are going to start looking at what is called a dihybrid cross. And this die should sort of give it away. So it means we want to look at two different traits. So we set this up when we want to look at two different traits. First, we need to figure out what our parents' genotypes are. So for two separate traits, so we're going to have two different letters representing the two different traits. And again, there's always going to be the dominant versus recessive alleles represented by the capital versus lowercase letter. Now, the first thing you need to do, like I said, is figure out what your parents' genotypes are. And then you need to write them in this notation. So one of the traits represented by a certain letter, and then the other trait represented by a different letter. As you can see, mom and dad have two different types of traits. And then you're going to FOIL the genotype within the mom or dad's trait, so within the parent's trait, in order to get the different combinations of these traits being expressed. So unless they are linked together, unless the problem tells us that they are linked together, that they're on the same chromosome, you're going to assume that big F doesn't always have to go with specifically one of these smaller b's for dad. It can go with either of them just as likely. So there's no preference for which allele of the other type it's going to go with. So you end up with all these different types of combinations of the two traits, and same thing for mom. So then you set it up just like you would for a normal Punnett square cross, but this time we're going to have one of each type of allele. So one of each of our little combinations here gets its own row and its own column. So as you notice, these two are the same for dad, and these two are the same. And same thing for mom. We got these two being the same, these two being the same. So you can save yourself some time and only have one, but let's, I don't want to confuse us too much on our first one. So let's just FOIL this. So then you do the exact same thing for our other crosses. You bring, okay, so for this box, we have this column intersecting with this row. So then you would take the F from this column, the F from this row from mom, and same thing with the B's to get the same cross. And since this is the same exact column, we're just going to write the same exact thing. There we go. So as you can see, you're taking the F from this column, so forth, so on, and same thing for all down here. Then you do the next one. Okay, so when you're all set and you're done with this, so you have all your crosses, your potential crosses that you can get from mom and dad, and when it asks about what is the percentage or the likelihood that we'd get all recessive traits being shown, you would count up how many out of these 16 boxes that would give us recessive traits. So for that example, you would say, oh, okay, we got 1, 2, 3, 4 that are all showing exclusively recessive traits. So that's 4 out of the 16, which then can be reduced to 1 out of the 4, which is 25%. So you guys pause me as needed to write this down for yourselves. You should be following along on page on to page 12 of our packet, so you should have completed that. So pause me or rewind me as needed to make sure you have this filled out for this bottom box, just so you have practice filling this in before we try it, and then you're on your own for it. Okay, welcome back. So now we're moving on to page 13 of our packet. We are going to do some practice of some dihybrid crossing. So up here we have the specific genotypes and phenotypes. It's actually all given for you, so both the homozygous and the heterozygous, which is pretty generous, especially now. So you guys are given the genotypes and the phenotypes that they're connected to. So we're going to do one problem together, and then you guys are going to be on your own for the rest. So number one, if a woman who is a non-PCT taster, which is recessive, which it tells us, and then you can also look up here and see that it's recessive, with a heterozygous hitchhiker's thumb, has children with a man who is heterozygous PTC taster with straight thumbs, which are also recessive. What is the probability of them having each of the following children? So it wants us to fill in the Punnett square. Bless me. And then answer these questions. So first, let's go. Well, first, let's pause it and see you guys try this on your own. So pause me now and complete 1A through E on our page 13 of our packet. Welcome back. So by now you should have completed question 1, A through B, the best of your ability, and then we're going to do the rest together. So first we got to figure out, it's asking us what our parents' genotypes are. So mom, it tells us, is the non-PTC taster. So we see that that's TT up here. So we're going to write TT for mom. And then she's heterozygous for hitchhiker's thumb, which we see hitchhiker's thumb, and heterozygous, meaning she has one of each type of allele, one dominant, one recessive. So those T's actually look, so T, T, and then H, H for mom. I'm gonna use a different color for dad. Let's use this pink. So dad, it tells us, is heterozygous for PTC taster. So we see up here that that would be TT, big T, little t. And then that he has straight thumbs, which is recessive. So 2 lowercase h's. Okay, so that's awful to see, mom. But so now we're going to set up our cross. So for mom, we see that she only has little t's to contribute, and then big H and little h. So we're going to put T, H, T, H, T, H, T, H. And then for dad, he has 2 different alleles for the T, and then only 1 H combination. So he's going to have T, big T, little h, big T, little h, little h, little h. So now we can do our cross. Let's do that in an even different color. Let's do that in this blue. So then we perform our cross. So we see dad has big T. It's supposed to be a big T, H, H, big T, H, H, big T, little t, H, H, big T, little t, H, H. And then same thing. Big T, little h, little h, big T, little h, little h. Okay, and then 2 little t's, H, H, 2 little t's, H, H, 2 little t's, little h, and then 2 little t's, and then H, H. 2 little t's, big H, little h, 2 little t's, big H, little h, and 2 little t's, little h, 2 little t's, h, little again. So, now see, we don't even get to see that. Okay, so now what we're going to see is, so now it's asking us how many PTC taster and hitchhiker thumbs do we have? So PTC taster is going to be homozygous dominant or heterozygous. And then also having hitchhiker's thumb, so big H, homozygous or heterozygous. So we see that we have one here, not one there. Oh, well, one here. No, just kidding. We don't have one there. One here, one here. And then I think that's all we have. So we have 4 out of 16, which is 1/4, which is 25%. Okay, so you guys please be following along, correct me if I'm wrong. So now for how many PTC tasters and straight thumbs do we have? So we have 1, 2, 3, 4. So we have 4 out of our 16, which is again is 1/4, which is equal to 25%. 25%. And then how many non-PTC tasters and hitchhiker thumbs do we have? So we have 1, 2, 3, 4. Great. So we again have this 4 out of 16, which is equal to 1/4, which is equal to 25%. And then we have our final question, which we're going to do in orange, I guess. And so it's asking us how many non-PTC tasters and straight thumbs. So that's just asking us for how many of our completely recessive traits do we have. And again, that's 4 out of the 16, which is 1/4 or 25% as well. I'm not going to write that to make it even worse. And then it's asking us what is our phenotypic ratio. And phenotypic ratio basically is just asking us what is the ratio of each of our phenotypes that we're seeing. So you can basically just get this from the answer that you just wrote. So we have 4 for our first one, 4 second, 4 third, 4 fourth. So it's going to be 4 to 4 to 4 to 4. And I'll write that out for us right now. So it's 4 to 4 to 4 to 4, which you can simplify to 1 to 1 to 1 to 1 because they are all 4, so they can all be divided by 4 to 1. And ratios are always going to be separated by colons, like this. Okay, so pauses as needed. Please ask me questions if this is confusing. And then, yeah, we'll move on. So you guys can do the rest on your own. So complete pages 14 to 17, and then pause me now to do that. And then I'll have the answers up later today, probably around 5 o'clock. And yeah, you're all set. So complete that now. Okay, welcome back. So by now you should have completed 2 through 4, and then this final page of your packet. So you should have completed up until page 17. So you're all done with your packet. Good job, you guys. So next week, just a little look ahead, we're going to be working on our mini QPA. You have, as always, our video upload, live classroom on Tuesdays, video upload, live classroom, video upload. So it's all here for your convenience. We're going to be working on mini QPA 2. So make sure you guys are all set for that. As always, here's my contact information. Please contact me with any questions. Okay, let's stop this screen share. See you guys next time. Have a great weekend.